Right, just testing to see if you guys can hear us from UBC.
We can.
Okay, great. So we'll get started. It looks like people have settled in. Okay, welcome to the 2023-24 season of VenBug. My name is Will Xiao. I'm one of the faculty advisors for the Vancouver Bioinformatics Users Group. And just want to know that please do turn off your phone during the event. And also to remind you that the event is being recorded. So, but the camera is really aiming at the speaker, so should turn afterwards. So we'll double check. But if you, for some reason, doesn't want to be recorded, then maybe wear a mask or something. Okay. Yeah. The view that you see in that back video in this room is what's been recorded and what's being shown to the other room. Those two monitors in the back. Okay, so our schedule, this is the first year we moved to during the day as opposed to after hours. So just to, but the schedule stays the same. First, we'll have a quick announcement that we're doing right now, and then that will be followed by an introductory trainee talk. Today will be by Eric. And then with some questions, we're trying to keep that first part to just about 15 minutes. We'll follow by a feature talk for about 45 minutes, followed by questions. And then we have pizza and drinks outside. Will be served around noon. And you're obviously very welcome to stay and chat with each other. And we have the space until 1:30 or so. So the organizing committee currently is has 3 faculty members, myself, Amy Lee in molecular biology, biochemistry, and also Faraz Haj, who's a UBC faculty, and he's actually on the other side at UBC. And I'll explain how that works in a bit, just because it's new as well. We also have a large number of dedicated student volunteers, and actually, usually it's the student up here, presenting the speakers rather than myself. And so this is meant to be mainly a student-run event, and the faculty's role is really just to support the students and also to order the pizza, so so student won't have to carry the money, the fee on their credit card and so on. So our role is really quite minimum. It's really the students who will be doing most of the work. And also want to highlight that we have quite a few students volunteers graduated this year. So we're looking for additional volunteers, and we actually have quite a few volunteers at UBC that's mentioned they would be supporting that, but at SFU site, actually, we're looking for additional volunteers, and we have one here today, a new person today. And okay, we also have a full lineup of speakers for you for the rest of the semester. So I won't go over that now, but you can see their names and affiliations up there. And also to remind you that we do send out direct announcements about these talks through our mailing list. So subscribe to the mailing list if you want to get the message directly rather than through your departments or faculty. And yeah, you can also subscribe to our calendars for updates. Oh, and I should also mention we, as a community organization, we're open to suggestions for feature speakers. So you can use the form on our website to provide your thoughts on who we should invite for VenBug. So there's a long list of VenBug sponsors. First and foremost is the graduate studies in bioinformatics that have been supporting VenBug over the last 10, 15 years and continue to support us to date. We have Genome BC, also long-term support. SFU, Big Data Hub is a new sponsor in the sense that we are using the facility here. And and also same thing with the Michael Smith Laboratory. We very appreciate that they let us use the facility. And to mention that this year is the first time we're doing 2 sites at the same time using Zoom. And actually the last time we tried this was probably more than 15 years ago when the technology was much less mature. But we actually did have sites, a main site at UBC and also a remote site at University of Victoria that they can tune in and watch. So if this model works well for the 2 sites, we will probably expand to include additional sites. But you also want to balance between not having too many sites so people can actually show up and socialize. Right, so that's why we're starting with just the 2 sites for now. And we're also trying to limit online participation because you just don't get the full experience and certainly don't get the pizza if you don't show up in person. We also have received some support from Langara, and they have a bioinformatics undergraduate certificate or actually degree program that also will also welcome students from Langara to join as well. And we have some industry sponsors such as Stem Cell Technology, and we're seeking additional sponsors as we go. VenBug is affiliated with Montreal, Monbug, and also Torbox. And VenBug was actually set up in 2001, I believe, by Francis Willett, and who then went on to set up Monbug and Torbox. And we're affiliated with ISCB as well. So yes, so Zoom audience, you can ask question in the chat, and the site at UBC, you can unmute yourself, but we're trying to make sure there's less voice collision. So you try to ask your question in the chat if you can, and obviously if you don't have a computer in front of you, then unmute yourself and speak up. So yes, go ahead. So yeah, so we were trying to experiment not to do it as complete hybrid, just so people do show up here. But if there's circumstance that cannot be avoided, then we will be happy to share the Zoom links and so on. So, and there are quite a few people on Zoom this time around as well. But yeah, we'll try to encourage people to start socializing in person rather than stay at home and tune in. Okay, so first I'd like to introduce Eric Navarro, and he's a graduate student in Keegan and Michael Kober's lab, and he's going to tell us about his work on Raman, our package for modeling the genome and exposon contribution to DNA methylome variability. And I actually remember Eric from the special topic course, being a very good student. So I'm looking forward to this talk. And I'll introduce the feature speak after.
Okay. Hi, everyone. Thank you for being here. And I'm very excited to show some of the work that I've been doing about tools that I've been developing called Raman, which is a package to model the genome and exposome contribution to DNA methylation variability. So I will start talking about DNA methylation. DNA methylation is an epigenetic mechanism defined as the addition of a methyl chemical group to the DNA, usually in a CpG context, which means that it's a cytosine followed by a guanine. So this biological mechanism is involved in many processes that regulate the activity of the chromatin. Some of them are gene repression, maintenance of heterochromatin, splicing, among others. So studying DNA methylation variability is important because differences in DNA methylation can be associated with differences in phenotypes in a human population. And these different characteristics can be sometimes associated with phenotypes that are of medical relevance, such as different disorders. And here, when I talk about DNA methylation variability, I am talking about the spread of DNA methylation levels in specific loci in a human population. So this variability can come from many places. Two of the main sources are the genotype and the environment. The genotype can affect DNA methylation through a mechanism called MQTLs, which describes the association of specific genotypes with DNA methylation levels, as you can see here in this figure. And on the other hand, the environment can affect DNA methylation through different mechanisms. And there are a lot of examples that have been described. One of the most popular ones is smoking. So the question of my project is, what is the contribution of the genome and the exposome to DNA methylome variability? So I want to understand if this variability is explained mainly by differences in the genotype, mainly by differences in the environment, by a joint effect independently of genetics and the environment, or by an interaction, which would mean that the effect of the environment on DNA methylation depends on the genotype that is underlying of that person. So the source of this DNA methylation variability, I think you cannot see the titles, is beginning to be understood. There are a couple of papers that have recently tried to explore this question. And what they have found is that it's usually a combination of the genotype and the environment, what describes better DNA methylation variability. So when this came out, was kind of a surprise for the field, because the meaning of epigenetics, like epi means above, and genetics is genetics. So this means that people thought about epigenetics as a mechanism that is independent on genetics, and that it didn't matter the differences of the genotype. So yeah, seems like even though the environment plays a big role and is involved in many of these models, the genotype also seems to be present. However, there are some areas of improvement in the field. One of them is the definition of the variable methylated regions, which is the unit that the field usually uses to measure DNA methylation variability. And I will dive a little bit more into detail how that can be improved. Also, because I'm playing with the methylome and the genome and the exposome, these are just like very data sets that are very high dimensional. And because of that, I have a lot of variables. I have a lot of CpGs, a lot of SNPs, and a lot of environmental variables. So this comes with many problems when it comes to modeling, which is something like called the course of high dimensionality. So because of that, I'm implementing embedded variable selection so that we can identify the variables that are potentially relevant, and then reduce the computational burden and improve the statistical models. Also, these previous papers usually describe the models that better explain variability. However, there are not any approaches to explore which models are actually better than what we would expect by chance. And finally, there is not a reproducible, easy to use method. So we know that in bioinformatics, it's not the same having the method in the paper than actually implementing it, right? So the method that I'm proposing has three main stages. The first one is to identify probes that are highly variable, that are the probes that we are interested in studying. So using variants, I first identify for a cohort or population, the probes that have high variability, and then I cluster them in something that I call variable methylated regions. So here is what I'm going to talk about the improvements, well, some of the improvements of my method. So usually, like historically, what has been done in the past is that they define a VMR as two or more probes that are contiguous, that are close together, and that they assume they are correlated. right? So one of the things that I'm implementing is a filter to check the correlation when I define these VMRs, because it has been reported before that when two CPGs are close together, they have a higher probability of being correlated. However, it's not the case for all the time. So it's not really something we can assume. So I'm doing that. And another thing that I am improving is that I'm taking into account the design of the platform that is used for measuring the DNA methylation. So if we only keep the definition of a canonical VMR, we would assume that we have a very good coverage of the whole genome, which is not the case for the field, because in human populations, the most cost-effective method to measure DNA methylation is using microarrays. And microarrays have a very specific design, so they don't target all of the CPGs. And for instance, one of the most used arrays is called EPIC. And that array targets a lot of regulatory regions by only using one probe. So by definition, if I just were to use this definition of VMRs, I would just throw away all of this information that is very valuable. And also, it has been documented that the probes that are designed to be there are very good representatives of the whole region that they encompass. So I'm creating this new category so that I also capture the information of those regulatory regions. So after that, I summarize the DNA methylation level of the whole VMR. And then the third stage involves G and E contribution modeling. So first, I do a variable selection step, which is a strategy that is based on lasso, so that I can select the variables that are potentially more relevant, and then I can move to feeding the models and then select the ones that are the best one for each VMR, and then characterize these models. So all of this methodology is encompassed in a method in our package that I'm developing that is called RAMEN. That stands for Regional Association of DNA Methylation with Environment and Genotype. So RAMEN has 5 core functions that you can see in this workflow how each of them works in the pipeline. And all of these functions are compatible with parallel computing, which is something very important due to the high dimensional nature of my problem. And also, all of these functions are very flexible for argument changes. And also something that is important to highlight is that the intermediate outputs of all of these functions can be used for other analysis. So for instance, if you have a DNA methylation dataset, you can conduct variable selection using the approach that I implemented. You can also, for example, many people in epigenetics want to do EWASs, which is epigenome-wide association study. So they associate specific environmental exposures with DNA methylation levels. So you can use my approach to reduce the number of probes or regions that you're testing and only focus on the ones that are highly variable, that are more likely to be the ones that are important. So reduce the burden of the multiple hypothesis testing. Also summarize regional DNA methylation levels of any region of interest, among other stuff. So I applied RAMEN to a case study, which is the child cohort. So the child cohort is an amazing study from Canada. So it gathers families from all over the country, and they follow the families for around 10 years for now. So it's a really, really impressive study. And in this case, I am using the samples that were collected at birth. So I'm using around 700 cord blood samples that have been genotyped using the Illumina GSA array and profile for DNA methylation using the EPIC array. So using this cohort is also an improvement for the field because previous studies have used cohorts that have a sample size of around 250 samples. So I'm having almost tripling this number. And also I'm using the EPIC array. And the previous papers use mostly the 450K array. And the EPIC array has around 850 probes, 1,000 probes. So it's double the size of that. And on the other hand, a very amazing thing about child is that it has a very good characterization of the environment, which is something that is really, really rare in cohorts. So previous studies have only collapse the environmental, like the exposome to 10, 12, and 19 variables. So this is not the best way to capture the complexity of the environment. And with child, I have 94 environmental variables that fall in 4 dimensions: parental psychosocial, maternal health, built environment, and nutrition. So this is also a cohort that is very interesting to explore. So after applying the whole method to the cohort, I saw that the additive model, which is the G plus E, is the one that usually better explains DNA methylation variability at birth. And something that my package does as well is that after it identifies the best model, it also conducts an analysis of variance decomposition, so that you can actually observe how each of these factors, how much variance is explained by these factors in all of the models. So we can also see here that depending on the model that wins, it can be environmental model, genotype, additive, and interaction model. And here on the left, you can see all of the factors. So for example, for the environmental model, there's only going to be a distribution for the E because it's the only factor that is playing. For the G plus E, you can have the variance decomposition of the G and the E, and for the G by E, you have for both of them. So the point here is that you can see that also the genotype in all of the cases and in all of the scenarios, the genotype seems to be explaining more variance compared to the environment. So I will finish this talk, concluding that additive models seem to be more likely to explain DNA methylation variability. And also the genotype seems to have a bigger influence in this scenario, specifically in the prenatal stage. Also that the package produces a reproducible method to conduct gene environment DNA methylation analysis. And something that I really want to stress out is that a lot of studies in the epigenetic field just associate DNA methylation with environmental exposures, and usually genetics is really kind of ignored. And as you can see here, genetics actually play a big role in DNA methylation, and it explains a big chunk of variance across the genome. So as a field, we should push forward to including genetics in all of the DNA methylation studies, because the environment plays a role, but it does together with genetics. So that's all. Thank you so much for listening. And thank you for everyone in both of my labs. And specifically, thank you for the families in the child study, because I could not be doing all of this amazing science without all of them. So yeah, thank you so much. And yeah. Oh, yeah. Yes. All the, can you repeat the question? Yeah, so the question was if all of the variables that are in child were considered environmental variables. So the question is, the answer is no. The child cohort has a lot of variables that were collected, and many of them are outcomes. So I manually created, like I saw the variables that were there, and I just included the ones that are count as an exposure. And also many of them are very indirect in specifically here because the baby is like in the uterus. So many things that happen to the mother can be like too indirect. For example, we have data for the father, but that doesn't really affect the environment of the baby. So yeah, so I conceptualized what the environmental exposure was. And then based on that, I selected the variables that were included in the study. Yes. Oh, yeah, the question here. Yeah.
So I just uh, can you hear me?
Oh, yes. So there's a question going there. Here. Oh. Yeah, so the question is if there was any methodological difference in how the environmental variables were taken in the cohort. So for all of the sites, they have a protocol so that it's kind of like standardized of like how they collect the variables. For some stuff, it's easier. Like for example, for diet, they have like the same questionnaire, so it's very consistent. It's the same way that the things are analyzed. For everything that involves health and, for example, psychosocial questionnaires, all of these questionnaires are questionnaires that are validated, and it's the same for all of them. So in all of these cases, it's very standardized. When it involves human contact, there's a protocol that kind of like tells them what to do. However, there's always variability in how people perceive. However, yeah, most of the things are really like made through questionnaires, so it should control at least some of that. Yeah, you're welcome. Oh yes, from UBC there was a question.
It was a short question. I just wanted to ask which kind of environment variables that you've used.
Yeah, so so the environmental variables that I use fall in 4 dimensions. So I'm using 10 regarding parental psychosocial. It involves parental maternal depression, involves life's adversity of the mother throughout the life. It involves the dynamic of the couple when the mother was partnered, and all of these kind of things, and also socioeconomic status, which is a combination of education, income, and all of that stuff. For maternal health, it's mainly conditions during pregnancy, which is infections and many of those things, and also something like smoking. For nutrition, is mainly the eating patterns of the mother 6 months prior to like during pregnancy. And I have different indicators of that, such as the healthy eating index, some measures of metabolism, dietary mediators that are involved in the pathway of DNA methylation, vitamins and supplements. And for built environment, that I have 9 variables, it is related to the outer environment that is man-made. So exposure to pollutants, the weather, child opportunity index, among other stuff. Yeah.
Thank you so much.
You're welcome. Yeah, so thank you so much.
We have one more question over here.
Okay, go ahead.
Hi, I just wanted to ask, can you hear me?
Yes.
Okay, I just wanted to ask, how did you record the levels of PM2, SO2, and NO2 for the built environment? Did you put like recording devices at the homes of the children, or how was that measured?
Yeah, so specifically for this stage, so we used a database called Canu. And so based on the postal code of the person, we identify the house, and then we have recordings of, well, like there's a database that has all of these measures of all of these pollutants in that region. So based on that, we link the place where the person lives to all of these recordings. However, for other age points that I'm working on, which is after birth, there is actual collection of dust in the house, and they like analyze the dust and see a lot of different exposures there that is more specific. So yeah, I'm working on that, but yeah, that's how it's measured.
Okay, thank you.
Okay, so we'll move on to our featured speaker today, Dr. Keegan Korthauer. She's currently an assistant professor in statistics at UBC and also an investigator at the BC Children's Hospital Research Institute. And her training, she did a PhD in University of Wisconsin Madison, and then postdoc at Dana-Farber Cancer Center and also T.H. Chan School of Public Health at Harvard. I guess then you came here afterwards. Okay, yeah, and then she started her faculty position here at UBC. Here in Vancouver at UBC. Okay, her research interests lie at the intersection of stats and biology. I think many of us are as well. And her group focuses on developing novel frameworks and rigorous inferential procedures that exploit the increased scope and scale of high-throughput sequencing data with the ultimate goal of uncovering new molecular signals in cancer, child health, and development. And it's a pleasure to have her opening our season for us. And her talk title is Methylation Matters, Tool Development for Community Discovery. And I want to apologize, I double booked myself, so I need to run out halfway through. So.
Thank you. Thank you for the kind introduction. Perfect. All right, so thank you to Will and the other organizers for the invitation to speak here. I'm really excited to talk to you guys about my work. And thanks to the VanBug volunteers also for keeping this running. All right, so I will skip the introduction there. All right, so before I begin, I just want to acknowledge that my workplace and my home are both located on the traditional and ancestral territories of the Coast Salish people, including the Musqueam, Squamish, and Tsleil-Waututh nations. And this is really something that I acknowledge as a white settler who comes to these lands and benefiting from an unearned privilege of living and working on these unceded territories. All right, so we've had a bit of an introduction to my research. So a little bit more specifically, the things that I get excited about are really untangling variability in genomic and epigenomic signal in particular. So this talk is going to focus largely on DNA methylation. So this heterogeneity can be differences between individuals, between whole organisms. It can also be differences amongst single cells in different tissues. And what I want to do is attribute these types of variabilities to things like changes in the environment, as Eric alluded to, or changes in different cell types, different stages in development. So all sorts of extrinsic and intrinsic signals. So that's really, really broad. So another way to kind of characterize my work is it's really a blend of sort of equal parts of working on the development of statistical models and computational tools to get at these things, and also then the application of those tools to actually infer the exciting biological discoveries. So it'll help me to tailor the talk a bit to at least the people in the room, because I can easily ask you. So if you have to categorize your interest into kind of the development of bioinformatic tools and or the use of the bioinformatic tools to make a biological discovery, how can I see a show of hands? How many are interested in the development of tools and or the application of the tools? And you can raise your hand for both. Hopefully you raise your hand for at least one. Otherwise, maybe we can chat afterward and I can learn about your interests in the field of bioinformatics. So how many are interested in the development of tools or aspire to be a developer of tools? Awesome, so lots of you. How many of you are interested in the application and making the biological discoveries? Okay, so lots of people are sitting at this intersection as well, so that's great. So this talk will be relevant, hopefully, for you. So in thinking about what I wanted to talk about today and in this seminar series, so the name of this seminar series is the Vancouver Bioinformatics Users Group. So it's inherently going to have some people that are using bioinformatic tools. And so I wanted to focus, even though my research splits pretty evenly this span, I'm today going to focus mainly on the tool side, development of the tools, a little bit less focused on these are the exciting biological discoveries. And in that, one of the themes I want to touch on is sort of the life cycle of bioinformatics software development as I see it, and some important things to consider in that sort of process. And then along the way, I also want to talk about a few of the broad main challenges that I think define the landscape of developing tools for analyzing genome scale DNA methylation data. All right, so just to set the stage, one of the most important things about developing bioinformatics tools that I really subscribe to, and if you want to read more about this topic of the question of should I create a new method, this figure down here, see if this pointer works. This figure down here is from a really useful blog post by Kieran Campbell, who did his postdoctoral work at UBC, is now at Toronto, but he has a great blog post that kind of has a philosophy of making that decision. Should I develop a new tool or should I just use a tool that already exists? And a lot of the philosophy that's outlined in this blog post is aligning with my philosophy as well. And so a couple of the two key questions that I think about when I'm starting a new project or thinking about starting a new project is first defining what is the problem or what's the limitation of the previous approaches. So if there are existing approaches that could answer your question or do your task, you should start there. If they can answer the question, you should use them. But if you found a problem, so this problem could be quite wide ranging. So it could be something that to do with the speed of the approach. Maybe it's too slow. It could be something to do with the accuracy of the approach. So some sort of problem that you've identified. And the second main question is, if you've identified a problem, maybe you have an idea of how you might solve it, you need to think forward to think about whether you can evaluate that you've actually solved the problem or not. And so that's aiming for this sweet spot right here in the intersection of all things that you could do that are feasible, as well as things that are important. And so that might seem really obvious and simplistic, but even for me, it's easy to kind of fall into something that ends up over here or something that ends up over here. For example, if I think of a really cool statistical model that I think would be really fun to implement, but it doesn't quite solve an important problem, or it doesn't give you a really different answer than something that previously exists, it's definitely not worth my time to develop that. There's some argument that could advance statistical methodology in general, but for my research, I want to solve a real problem. So I think these are things to have in the forefront of your mind, and they'll kind of call back to these questions as I go through. All right, so I'm not going to introduce DNA methylation. We've had a nice introduction from Eric. So I just want to kind of remind, frame the thinking of analysis of genome scale DNA methylation data and the importance of statistical modeling and rigorous inferential procedures. So this is a quote from a 2018 review on sort of the analysis of an integration of DNA methylation data in general, ranging from all different types of ways you can measure it, arrays to sequencing. And the quote is that rigorous and reliable inference of DNA methylation data is key to a wide range of downstream tasks. So that really describes kind of why I do what I do, which is focus on the statistical modeling. So the broad challenges, as I see it, and the ones I want to discuss today of analyzing genome scale DNA methylation data are paying the price of multiple testing is a big one because of dimensionality, actually defining the features and selecting the features, computational efficiency problems, as well as the fact that when new technologies arise, they often demand new methods to analyze them because of new biases they introduce or artifacts. So for the rest of this talk, so I want to go through these challenges and talk a little bit more about each of them, as well as along the way highlight some examples from my work. So in the first example, I'm going to talk about the process of developing a tool kind of from that whole cycle, where the cycle is kind of, it's done in iteration. So starting from being motivated by a community discovery, identifying a problem, proposing and evaluating a solution, implementing and disseminating, and then back to leading to community discovery. So this example is now getting to be a few years old, but I think it's nice in that it illustrates that sort of tool development process. And then I'm also going to talk about getting into the fourth challenge, some ongoing work in particular that's dealing with newly available experimental techniques. All right, so the motivating example is dealing with this problem of trying to establish correlation versus causation, or get at causation. So we know from observational data that when we look at measurements from the same samples of methylation and gene expression, that we can often see these negative correlations if we look at the methylation level in the promoter region of a gene, which is just upstream. But the key question that's not really solved is whether these associations that we can see, so this is observational data, and the fact that we see more points in the lower right and the upper left suggests that the predominant relationship here is that when methylation increases, expression decreases, and vice versa. So certainly there's counterexamples. And the question is this just a result of other things that are causing this, or is this a causal relationship? And a study from 2017 asked this question for the first time in a rather genome-wide answer. Previous studies up until that point had targeted that question, but kind of one gene at a time or a handful of genes at a time, and kind of found mixed results. And this study generally came to the conclusion, so this is a quote from the paper, that promoter DNA methylation is generally not sufficient for transcriptional inactivation. And so I don't have time to go into the details of what they did, but essentially they took advantage of a zinc finger system that is doxycycline inducible, and were able to forcibly methylate the promoter regions of thousands of genes. And this is a figure from the main results of their paper, showing that as methylation increases and looking at the change in expression, that even as you increase methylation further and further and further to the right, there's still a lot of points that are increasing in expression. So this ultimately is what motivated their main conclusions. So why are we talking about this example? So this is kind of the community discovery that motivated the thinking of maybe there's some room to improve here. So looking at this result, this might be, for me, a little bit surprising, expecting to see maybe a stronger correlation. So the question is here, how is this data analyzed? And if I analyzed it differently, would I come to a different conclusion or not? And so that was kind of the question I had. So in thinking about developing a new method, this data was analyzed with what was a state-of-the-art tool at the time. It's still a very widely used tool, particularly for single CpG site differential methylation analysis. So why do we need to develop a new method? So thinking about what is the problem and what's the limitation. So the thing that might give someone pause, and the conclusion of that paper was a bit provocative, is that there were surprisingly few, comparatively few, and relatively weak associations between the methylation increase and the expression decrease. And so in thinking forward of, okay, if I develop a different approach, would I be able to detect more associations or stronger associations? So that's kind of thinking forward, because I don't want to start down this road if I don't have anything to evaluate. So thinking in this way is good at the start of the project. So this is a bit of revisionist history, as I was already working and already developing the method. But I had, at the time, been working with evaluating on sort of observational data, looking to see if I could improve and detect stronger correlations in that. And then when this data set became available, it became a stronger use case. But definitely, this is the evaluation question is always something to think of as you're starting the project. Okay, so moving through, I want to now talk about the main challenges, the broad challenges that I see in the analysis of genome scale DNA methylation data. So to help kind of frame my emphasis of this talk, it's going to be helpful to know how comfortable everyone feels with kind of statistical analysis, p-values, multiple testing, false discovery rate. Can I see a show of hands if you feel I really understand what false discovery rate is and why it's necessary to control for multiple testing? Okay, so if people feel pretty comfortable, I'm going to guess that the rest of you maybe could use a little bit of a refresher. And yeah, that's not anything to be ashamed of. It definitely helps to look at these things more than once. So even if you've learned about this, sometimes it isn't always in the forefront of your mind. So what are the two types of errors that we can make in statistical testing? So there's the type 1 error, that's the false positive. The type 2 error is the false negative, so we didn't detect something that's there. That's bad, but oftentimes we're more concerned with making that false positive and saying, okay, this is an association when it didn't actually exist. So if we think about our classic statistical test, our 0.05, that's what we're generally concerned with. If we don't consider correcting for multiple testing, and we just have our 0.05 threshold, we do lots and lots and lots of tests. Why is that a problem? So this is kind of showing it with the math on the left, which you can study on your own if you'd like. But essentially, if you plug that into the formulas here, if you have a function of how many tests you're performing, and on the y-axis here is the probability you've made at least one false positive error, one type 1 error, you can see that once you get on the scale of on the order of hundreds of tests, you're virtually guaranteed to have made at least one false positive. So this is the problem of multiple testing. It's why we need to care about this problem. So in classical statistics, we would do things like control that error rate. So if you are in the field of GWAS, you might be familiar with Bonferroni. Super conservative procedure lets you control that probability you've made at least one type error. In genomics, that is generally not thought to be as practical. We don't necessarily need to be so strict about controlling the probability we've made at least one error. And we're usually a little bit more willing to say, we can make some errors as long as we can control the overall total proportion of errors that we've made out of our discoveries. And that's where that false discovery rate comes in. So in genomics, we often have many more than hundreds of tests. So an example that's relevant a little bit more for transcriptional space, for humans, if we have 20,000 genes, say 500 of those are truly differential. If we optimistically had 100% power, that means we're going to detect all of those true false positives. And if we have that 5% type 1 error rate, we'd make a lot of false discoveries. So you can see in this best case scenario, if we don't control for our multiple testing, just driving this point home that 66% of our results are false positives. And the problem's going to get worse as the proportion of null increases and the power decreases. So this is the sort of what was at the time a bit of a radical idea, saying it's okay if I'm pretty sure I'm going to make false discoveries, as long as I know I can kind of control that proportion of my overall discoveries that are false. And that was proposed by the Benjamini and Hochberg, the FDR correction procedure. FDR control is actually an active area of research in both how you do it and other extra information that you might be able to incorporate in controlling that rate and maximizing your power. So this is an example figure from a paper of a benchmark study where we kind of compared head to head a bunch of state-of-the-art methods that were coming out all around the same time to see how useful they would be. So getting back to DNA methylation data, which is what we're primarily focused on here. So why is this so particularly challenging in DNA methylation data, genome scale DNA methylation data? It's simply the dimensionality. So in the human genome, for example, there's close to 30 million CPGs, so 30 million sites at which we could have a DNA methylation measurement. So in whole genome sequencing, bisulfite sequencing, we get a measurement potentially for every one of those sites. And then in other types of data, such as array, we still could get on the order of hundreds of thousands, close to 1 million sites. So it's much more high dimensional than, say, transcriptomics experiments, where we have on the order of 20,000 genes. All right, so what else makes DNA methylation, genome scale DNA methylation data so challenging to deal with? So the process of actually defining what features you're going to use and then selecting the important ones is also a little bit unique, or a little bit different in DNA methylation compared to if you're more used to thinking about transcriptomic studies. So this is kind of a data matrix image, more relevant to transcriptomics in terms of dimensionality, 20,000 genes. So again, in DNA methylation data, if it's whole genome sequencing, we could have 28 million CPGs for human. So what do I mean by feature definition? So we have these measurements at every CPG for sequencing, or every probe, which represents a CPG in array data. And so we could look at those measurements one at a time for each CPG, and we would get a level. Maybe we have different groups in our experiment, and we want to know, are those CPGs differentially methylated by those groups? So the task here would then be, if I've defined my features as individual CPGs, my selection task is then to pick out the CPGs which are showing differential methylation. So that's one option. And I might do that across all of the CPGs I have measured. So this could be hundreds of thousands to millions. And I would pick out these sites. Another option is I can define features as groups of CPG sites or regions. In that case, the thing I want to detect is regions or groups of CPGs that have different methylation levels according to some condition, or variably methylated regions that have heterogeneous methylation levels across all samples, as was relevant to Eric's project. So here, the definition task, sounds straightforward. I want to group these CPGs together, but how do I group them together? These sites are potentially every cytosine that's followed by a guanine is potentially methylated in the human genome, as well as non-CPG methylation, if we want to consider that, which is relevant in neuronal tissues and other organisms. So how do we group them together? They're non-randomly spaced throughout the genome. They don't naturally group together in, for example, genes, although you can annotate them as being in certain genic regions. And then the second question is once I've defined what a region actually means, how do I pick out the ones that are actually meaningful and significant? And so the main considerations for deciding, do I want to define features as CPGs? Do I want to define features as regions? Is how am I going to interpret those biologically? What is it going to mean for the dimensionality of my analysis task? So obviously, if we're looking at CPGs, that's going to be many, many more tests than it is for regions. On the other hand, for biological interpretability, it's been argued that with the exception of certain CPGs that have been termed CPG traffic lights, which kind of operate kind of independently on their own, oftentimes groups of CPGs have similar signal and can be maybe thought of more as the relevant functional unit altogether rather than acting independently. And this is also really tied in with the statistical assumptions that we need to consider if we're going to analyze these separately. So if we just consider them independently, ignoring any correlation, that might lead to some false discoveries, which I'm going to discuss, as well as how we're going to assess the significance. So if we wanted to consider these CPGs as testing them independently and then grouping them together later. So this is what I'm calling post hoc. So after my statistical significance assessment, I'm going to group them together and detect regions. Then what's going to be the problem? Well, so the reason I'm mentioning this is the approach used by the method DSS that in the example, the motivating example that I discussed. And it's also kind of the most common approach generally for analyzing DNA methylation regions. So the problem here is that in this example, in this cartoon, if I assume that I know the location, that there's an actually differential methylated region at this location, all of these light blue sites are true positives, but I've also detected some significant sites that were false positives. Then my false discovery rate, which mathematically is just the expected proportion of false positives out of the total number of discoveries. If I think of that on the CPG level, then only 2 out of 10 of my discoveries is a false positive. So that might be fine if that's what I specified at the outset of my analysis. The problem is if I'm grouping these together after the fact, which again is a common approach, and I consider this rate at the region level, then all of a sudden I have 1 out of 2 regions that I've detected that are false discoveries. And with this type of procedure, there's no way to control that or to even estimate it. So this is a major limitation of previous approaches and a challenge in general for the analysis of this type of unstructured genomic data. So a third challenge that we can't ignore is computational efficiency. So as I mentioned, DNA methylation data is very high dimensional. We have millions of CpGs in the genome. So things that I think about when I'm thinking about what type of approach am I going to target that's going to help me solve my problem is always going to involve this trade-off between feasibility and accuracy. So these are just a few of the things that might push you in either direction. So if you need to push over into the side of speed, then you might be looking at things that are simpler models. They're using approximations that have analytical solutions, trying to get away with fewer parameters, even if you're not fully modeling the complexity of the system, as opposed to more complex, fully hierarchical models and doing things like permutation. So sometimes those things are necessary if you need them to gain accuracy, but it's always going to be a balance and a trade-off. And I want to just kind of skip ahead so we make sure we get through everything. So an example of this, a particular method development to try to improve upon the detection of these associations in whole genome bisulfite sequencing data is the approach that I developed called DMR-seq. So this is kind of moving into the step of, I've identified the problem. Can I use a different method that's going to identify stronger and more associations and use this case study data to evaluate it? So the features, kind of key features of the approach that kind of relate back to those key challenges are that it's region level testing. So we're not as hampered by the dimensionality because we only have one test per region, rather than testing each CpG at a time, one at a time, as done by the previous approaches. 2, the feature definition problem, in contrast to relying on predefined regions, we're detecting the regions actually from the signal itself by grouping things together based on their having similar signal. And to try to increase the computational efficiency, since this type of approach necessarily, as we've defined it, requires permutation to establish significance, is in the modeling step, instead of using full-scale count modeling to actually model the count nature, we use approximations to approximate the and get something that's much more analytically tractable. So this is a bioconductor R package that's available. So this is kind of the implementation and dissemination step, making this available. But I really want to get to kind of the using this approach. So these might seem like subtle choices and subtle changes that we've made instead of doing the significant CpG by CpG, grouping them together, and now we're testing on regions. These seemingly subtle choices, they can make a big difference. And this was kind of the question that we had at the outset. Does this make a difference? Do we see a difference in this same data? So this is reanalyzing the Ford 2017 data, but using the new approach and a similar structured figure where instead of absolute methylation level, we have the statistical significance level because we can get that now with this approach. And we see that as we increase in the methylation signal and we try to count up how many of those associations are significantly decreasing with increased methylation as opposed to increasing, that as we slide further and further, that the strongest or the most statistically significant associations are the ones that actually correspond to the strongest signals in the repression side for the expression. And to round this back, and this is, again, the reason that I chose this example to discuss today, to talk about this full circle. So after implementation and dissemination, it's getting back to this community discovery. So this community discovery is kind of maybe not the best term to use, but so community discovery, I mean discovery by the community or also including yourself. So anyone using the tool making the discovery. So here in this example, this is a published paper that turns out to be the revised version of the original preprint. So this came out last year. And in this tool, you'll notice that instead of using the DSS software, they've now used DMR-seq and come up with a very similar figure. And in addition, they've changed the overall conclusions to promoter DNA methylation is not generally sufficient for transcriptional inactivation to now promoter methylation is not exclusively associated with gene repression. So again, it's a subtle change, but it's just acknowledging we're not going to claim we did not see this strong signal, therefore the repression is not necessary, but instead it's switching that conclusion to, okay, this is context dependent. We do see that association, but it's not exclusive. There are counterexamples. And then kind of within this cycle all throughout, there's this whole issue of software maintenance. So once you put out a tool, ideally you don't just leave it there and not touch it. So hopefully, you're getting people to use it. Those people are giving you feedback saying, okay, I ran into this error, or I thought of this feature. I really want to do this, but it's not implemented. Is that something you can do? And so there's a lot of back and forth with the community. And by being responsive to those inquiries, that increases the usability of your tool. And it's good for the community. You want them to be able to apply it. All right, so that kind of leads us through the first three broad challenges in that kind of case study of software development. So those three challenges are going to stick with us for any type of DNA methylation assay. But there's many types of DNA methylation assays besides sequencing, bulk, which is what I just discussed. And these new experimental techniques are necessarily going to have some different characteristics that we're going to have to factor in. And so in this example, I want to highlight some method development for analyzing variably methylated regions. So these are the VMRs that Eric introduced. But now instead of in bulk samples, we're going to be talking about single cells, measurements in single cells. So what is the challenge? Why do we need to even discuss this? Why can't we just apply DMR-seq? So one is that this data is inherently structured differently. So instead of each sample being a measurement of an average across thousands or millions of cells, now it's a measurement of an individual cell. So that data, that signal to a first approximation is going to be binary. So either the site is methylated or it's not. Yes, in a diploid genome, you could have hemimethylation, but it's relatively rare. And two, with the level of coverage that we have in current technologies, it's not reliable. We're not able to reliably detect that over background sequencing error. So to a first approximation, we have this binary signal. The second main feature that makes this very different from bulk sequencing is all of these missing sites. And so on the order in typical experiments of 80 to 95% missing, where we just don't see whether that site is methylated or unmethylated in that particular cell. But the trade-off to getting lots and lots and lots of cells. So the analysis task here is, again, we don't necessarily know much about each individual cell in terms of, so maybe we don't have groupings of cancer versus normal, or repressed, methylated induced versus unmethylated induced in the previous experiment, where we just have this group of cells that we know came from a particular tissue. And so our task here is to find regions of the genome that have variably methylated signature across all the cells in the experiment. And then we want to use those to do some downstream analysis tasks. So one thing that we would commonly want to do is then cluster the cells and see if we can recapitulate some types of cells or some cellular states. Okay, so for this, we want to go back to that first main question we need to think about, which is what is the problem or limitation? So if we're going to analyze this data, we can do a check. We see that there are some methods already available for analyzing single cell methylation sequencing data. So we go and check those out. So in particular, there's a bioconductor R package. There's also this SEBS command line tool. So what is the challenge? So again, the previous challenges remain. We have these multiple test inverting, the feature definition challenge, the computational expense. But we have this new challenge of this. Now we have really sparse data, and we have even larger dimensionality. These previously existing approaches try to tackle those challenges. But one thing that is in common with them is they rely on either predefined regions. So I'm going to go in and I'm going to look at promoter regions, or I don't have regions of interest, but I'm going to look at sliding windows or binning of some sort. And so the question we ask is, can we do better? Can we zero in and pinpoint with single site resolution where these regions are, even in the face of the sparsity? And the kind of key to doing this is, I'm going to unfold in this figure here. So as I said, each site is very, very sparse. We only have measurements on a random small subset of cells at each site. But if we zoom out and look kind of at the region level, which, as we've argued, may be the more relevant functional unit for a lot of biological processes, then we can look, what signal we get is we can average the methylation level across all cells. So in this cartoon, I've got a genomic region that has 70% of the cells methylated. And then outside of that, all the cells are not methylated. So the key here is that since the underlying state of underlying data is essentially binary, we can think of this as decomposing into two groups. There's a group of cells that have no methylation in that region, and there's a group that has methylation at that site, so the zeros and ones. So the key here is that we can always break that down when we zoom in, that there's two groupings. It doesn't mean that there's two subpopulations. We want to have an arbitrary number of subpopulations. So here it's illustrating how this could happen in the case of 3 subpopulations. So 2 of those happen to be methylated at that site or that region, and 1 is not methylated. But this could be an arbitrary number. This could be a much larger number. So say we had 18 subpopulations. We can always break it down into, when you zoom into that region, is that grouping, is each particular subtype in the U or the M grouping? And then when we use those features collectively for the whole genome, then they can be informative of cell subtype. And I want to speed through this because we're running low on time, but the key to the method, the way the method works is the first step has a lot of similarities to DMR-seq, but the way that we infer whether that region has evidence of having variable methylation is using a hidden Markov model to infer, the likelihood of that area having 2 underlying groupings or just 1. And then those groupings are then ranked in order of their evidence, in order of their probabilistic strength of having 2 subgroupings. And then with that list of VMRs ranked in order of their likelihood of having 2 groupings, then we can then recapitulate. So this is applying the method as well as comparing it to previous approaches on some data from Luo 2017 science paper of mouse neurons. Then we can compare the groupings when doing unsupervised clustering on some cell type labels. And the cell type labels here, importantly, are applied from non-CpG methylation. So it's not using the exact same data because that would be a problem. And so we see we're able to recapitulate the broad cell type labels in the dashed lines, as well as the finer subtype labels in the text, over and above in terms of a clustering performance metric compared to previous approaches. And then we want to think of other ways we can evaluate that. Since in this example, the cluster cell type labels, again, they're derived from the non-CpG methylation, but it is technically on the same cells or the same data type. And this is an example of some multi-omic data from a paper from 2019 in Nature of mouse embryonic development, which happens to measure both methylation and expression. So here we can actually look, is the association of the variable methylated regions of the methylation with the expression in promoter regions, where the promoter is an independent measurement, do we get stronger associations in the variably methylated regions compared to something more simplistic like predefined regions, for example, promoter regions, as shown in this figure. So here is directly putting them head to head. If they have the same strength, they'll be along the diagonal line. But if the VMRs have stronger signal, they'll be in the blue area. So you can see most of the points are in the stronger area, although that there is some broad agreement. as well as the VMR-seq regions also are picking up on some positive correlations, so in the inverse direction for when the VMRs overlap with the gene body. So that's something harkening back to the complex relationship between methylation and expression, that depending on the context, you might get different associations. All right, so this project is currently in development. So this is work led by a PhD student in my group, Ning Shen, and she's also developed an R package that implements that. And so this is part of the implementation and dissemination. And so this is something you can actually go check out on GitHub. So eventually we'll be submitting it to Bioconductor. So that this is an example of a project that's in this stage. You know, it's not quite submitted, but hopefully we round that back and we kind of continue the cycle with this project as well. Great. And so just to touch on a couple of other things going in the lab that are in various states. So you've already heard from Eric talking about his awesome work in modeling the contribution of environment and genetics in contributing to that variability in methylation. And he also was talking about his R package and nicely went through, you know, what are the limitations of the previous analyses and what is my approach going to contribute, and then making an R package that's going to let people with similar data answer these questions and see, okay, in different cohorts, do we see the same signal? How do things differ? Noting differences in, you know, as we were discussing with the questions and how people measure the environment, that might matter. And a final example is Giuliano's a PhD student in my group as well. So he's recently developed an approach for a Bayesian method to do decision curve analysis. So this at the surface seems very unrelated to what we've been talking about. So decision curve analysis, if you're not familiar, is a formal decision theoretic approach to evaluating, validating clinical prediction models. So if you give me some data about a patient, if it's an omics-based clinical prediction model, maybe you're giving me some biomarker information, and then I'm going to put that through my prediction model and then decide which treatment am I going to give. So we always want to evaluate those before they're actually implemented and compare them with, you know, if there's a standard of care already or alternative approaches like I'm going to treat everybody or I'm going to treat no one, and make sure that at the clinically relevant decision threshold, which is determined by clinicians, and it essentially weights the benefit of a true positive versus a false positive, and that's going to differ in different contexts. So is that new prediction model going to be giving me a net benefit overall compared to the standard of care or treat all or treat none? So previous approaches, you would just look in the classical decision theoretic approach, you just look, is my curve for the new model highest at that decision threshold or not? And then that guides your decision. What Giuliano worked on is developing an approach that gives you this uncertainty quantification. So given the data that I'm using to make this evaluation, can I have some quantification about how certain am I that this is really meaningfully greater in terms of net benefit than the existing approaches? Or is there a considerable uncertainty? Am I only 60% sure? Then that's going to guide whether I think I need to actually do some more research to really be sure that I want to evaluate it or implement it. So how this is relevant for this framework of methylation is Giuliano's now working on applying this methodology to evaluate the recently proposed methods for multi-cancer early detection tests that are based on cell-free DNA methylation data. And again, this is an R package that can be kind of generally purpose used for evaluation of clinical prediction models. All right, so with that, I'll not get in your way of eating pizza, but I'm happy to take some questions either here or offline. And yeah, thanks to all my funding sources, NSERC and the BC Children's Hospital Foundation, as well as CANSI, and thanks to all my awesome students in my lab. Yeah. Yeah, so the question is a CPG grouping, is that similar to looking at like CPG islands? So that is a way that you could group CPGs. So you can classify them as in islands. So that tends to be regions where there's a lot of CPGs. They're very dense, as opposed to regions like the open sea, where there's very sparse. So there's a lot of distance between the CPGs. So that is something you can use to kind of guide how you bin them together. I wouldn't necessarily think that that would be enough to give you your region definition, simply because maybe if you just wanted to look at CPG islands, it might work. But for shores and shelves and open sea, those can be quite long. And it might be difficult to argue that that whole region is a functional unit altogether. There's probably a lot of change that's happening within it. Great question. Yeah. There's a big conversation with community. And what I'm wondering is how is the funding? It's a great question. I think that's a problem that's being more widely recognized. Personally.
Excuse me, can you repeat the question for the UBC audience? Thanks.
Yes, so the question is, how easy is it to get funding to maintain your tools? Because maintenance of tool and their shelf life is important. So the question was, do I get funding to do that, specifically to maintain tools, or do I just do that on my free time? So I currently do that on my free time, but I am aware of some funding initiatives. So for example, the Chan Zuckerberg initiative has been supportive of giving funding directly for, in particular, I remember a funding call that was targeting someone who has a tool, but they need funding to keep it alive, do further development on it. That said, I think that's the only example that I can call to mind. So it's not a common thing. It would be great if it becomes more common. Yeah. Yeah, so the question is, given that there's a trade-off between the number of samples and the number of sites, is there a benefit to using cross-validation techniques? Yeah, so I guess the outcome of that would be that you would be able to better evaluate whether you're making false discoveries, because if you don't see it in the held out sample, but you saw something in the training and you don't see it in the test, then you'd be more skeptical. Yeah, so things like that will definitely help you for evaluation. I'm not sure that is going to help you. Yeah, no, I think it's a great point for evaluation. I'm not sure how I would use it because if you start using the information you get from the cross-validation to then inform the model fitting, that a danger of overfitting. But a technique like that, yeah, would certainly be useful for evaluating what you're finding. Like another technique beyond, I showed the example of comparing with labels that were derived as independently as we could from the same data set. And then another example, comparing to multimodal data. So it's like an independent measurement of a different thing that we think should be correlated with our signal. But cross-validation would be another good idea for something to evaluate. So if you have lots of cells, maybe you want to hold some out, do your discovery on most of the cells, and then see if what you find is applicable to the held out set. I guess the drawback of that is going to be a little bit limited in rare subtypes or rare cell type or cell states. You're going to be losing your power to detect those. And also, if you don't see them in the held out set, maybe your held out set was too small. But certainly for broad trends, it would be useful. Yeah. Yeah.
That was very good talk. In your single cell model, it was interesting that you had, I guess, binary emission for
Yeah, so it's not for the whole region. Maybe I can go back. So it actually is site level. So each site can have a different emission. You can have two groupings that are methylated, unmethylated, or one of each at each individual site. Yes. Oh, yeah. For each grouping. Yes.
So I guess that's the biological version itself. If method is fully once you take into account cell type, if you have a say about that.
Yeah, that's a very good point. So that is an assumption, it can be a strong assumption. So the question, yeah, sorry, I didn't repeat the question. The question is the assumption that the cell type or state that you're identifying, they all have the same methylation state within that region? That is an assumption that we're making, and it's a strong assumption. So I would say it's maybe more relevant to, so this kind of leads into the whole question of what is a cell type. So I think I prefer to call them cell states, and then it feels a little bit more comfortable to make that assumption that, maybe there's two groupings of a cell type, but they're in different states and they differ by this methylation level in this region. But yeah, that is a limitation, that if there is stochasticity with drilling down into the subtype level. Yeah, that said, if that exists on a very relatively small number of regions compared to the overall picture, then maybe it's not going to make that much influence. But if it's broadly occurring and it's super stochastic, then that's really going to hurt. Yeah.
Great. Thank you. And I'm going to take this moment to let some UBC questions come.
Hello, can you hear me?
Yep.
So I was really interested to hear more about the DMR-seq method. And I was curious what happens with this method if you have a few individuals that kind of are driving the DMR that you end up finding, or that like shift if not everything is, and if there's a way to account for population, I guess, on the individual level in that tool.
So it seems like you're wondering about what happens if you have kind of outliers in.
Yes, exactly. Outliers that might cause, I guess, the average to shift for a particular either CPG or DMR.
Yeah, so I guess it's going to be dependent on how many samples you're analyzing in the first place. So if you have,
right.
The smallest experiment you could have, but still have replication like a 2 versus 2,
Yeah.
That's going to be a different story than if you have 20 versus 20 and there's one sample that's different. So it's going to have comparatively less influence the larger number of samples you have versus, if you have just a couple, 2 or 3, that's going to be a large part of the signal that it's inferring. But also if it looks like an outlier, that means it's, if it's far from the rest. And in terms of the statistics, that means there's going to be a lot of variability, which means it's harder to get significance. So there is kind of a field of research that's trying to get at, instead of differential between two groups. So the goal of DMR-seq is to find which regions are different on average between two groups, assuming that there's not a ton of heterogeneity. But there's another kind of way that you could frame the problem that says, I'm going to compare maybe 1 or 2 special individuals with the rest of the population. And I have kind of like a 1 versus all test, kind of like outlier detection methods. And so if that signal is what you're interested in, then I'd encourage you to kind of look at those types of approaches rather than trying to average everything together if you know that there's differences and you're interested in those differences.
Okay, thank you.
Anybody else who'd like to ask a question here? I think we have space for one more question. So, do you want to go for it? Yeah.
So, can you hear me?
Yes.
Yeah, so while I was listening to your lecture, I was just searching online about on various machine learning or deep learning methods that can handle these sort of problems. For example, you told me that high dimensional sparse data is one of the concerns. So I was searching up on well, deep neural networks are known to handle high dimensional data very well, but I was just curious about if you're utilizing those methods or when I search online, it says that lasso and its variants can handle high dimensional sparse data sets.
Can you say the last what can handle the high dimensional sparse?
Yes, I'm sorry.
Sorry, I just missed the I think you maybe named a method, but I didn't quite catch it.
Lasso.
Lasso. Okay.
Yeah. But I'm not sure because I haven't dig deep into lasso, but I just searched online and said that it says that it could help. So
so those are great questions. So I think that it's on a couple of things. So you talked about deep learning and you talked about kind of machine learning, which they're highly related. So lasso is a machine learning technique that helps with feature selection. So that can be certainly relevant to this problem and is actually features in Eric's project in terms of kind of throwing a bunch of features in and then trying to downweight the ones that don't seem important and keep the ones in that you do think are important. So that can be one way to ameliorate that. That's not necessarily a deep learning approach. A deep learning approach is going to have these hidden layers and they're going to be a lot more complex and a lot of parameters to tune. But they certainly exist and they have been applied. So I'm aware, I mean, for the task of inferring, sort of imputing methylation states for your missing information, I think that's a great approach. So if you need to know the methylation level of a particular cell at a site that you don't have the data for because you don't have the data for most of the matrix that you are given data for, then the deep learning approach can help. So you could certainly go that route. We've avoided the imputation and simply taken the approach of, okay, let's look at the signal in aggregate and then see what we can get using that information in the best way we can. But you could take the alternative approach, which is impute the things that you don't have and then do your clustering based on the features you define. Yeah, that's a perfectly reasonable approach. I don't know if that helps answer your question.
Thank you so much.
I think that's all the questions, or do we have space? Do we have time, Rhiannon?
Grab the mic so I don't. Unfortunately, that's all the time we have. We have to clear out of this room on this end. But I'm sure there's opportunity over email to follow up in the future. Just on this end, if everyone can go to the atrium, there's now should be pizza and beverages ready and some chance to maybe ask some more questions. And then on the other end, just a heads up that you guys have the room till 1:30, need to be cleared out before then. And thank you everyone for coming. So thank you so much for your presentation and you as well, Eric, and just another round of applause for speakers.